Hello, Evo friends. I'm just going to give you a quick video so we can finish out this last chapter and spend all of Wednesday reviewing. And then I'll throw up a video if we need more review. Okay, so a review that we already did, right? We have shared characteristics. This is important because it tends to get confusing. People get confused sometimes, but everything here shares a characteristic, right? Who has a hinged jaw? Everybody above here has a hinged jaw. Everybody above here has legs. Everyone above here has hair. Okay? Yes? Yes? This is making sense? This is used to determine relatedness when constructing these cladograms. And they are shared characteristics. Okay, shared characteristics. Synaptomorphy. These arise within the larger clade. It distinguishes those that share it from those that do not. So those that have hair, those that are warm-blooded, those that are mammals, those that have a vertebrate. Provides information about relatedness within the larger group. That's what we use the clades for. Used to identify branch points within the larger clades. So this is what we're looking for. These guys are a derived trait. This is not. These are not. These are not related just because they're next to each other, right? These, we're looking for this kind of a relationship, okay? Okay, choose the right relationship. This is what we need to do. Okay, so we're looking for rule of parsimony. Parsimony, I've heard it both ways. Okay, so if you look at this one, it feels overly complicated. We have things here and things here. I don't like that. This looks better, saying, hey, this trait, hair, legs, whatever, makes sense here because both C and D have it, rather than trying to put it here like this. We don't want to do it like that. Using many characters, you should, to develop an accurate cladogram, often results in many possible trees, right? You will, as we keep doing this, you'll be like, oh, it could be like this, it could be like that. Ideally, you want it to look like this. So the simplest cladogram is the fewest steps or the fewest potential events. Okay, again, the concept of this started with Charles Darwin. He was on an island. He's like, hey, these things look like the birds on the mainland. Let's make a little chart and see if I can make it make sense. I just want the beaks to make sense. Since then, of course, we have new technologies. We have Evo Devo. We have people who do this nonstop all the time. So we used to think of fungi and plants were closely related, but now that we actually know that fungi are more closely related to mammals, to us. We also used to think birds were a separate class of vertebrates. Now we know they are descended from dinosaurs, which is actually cool. Again, dinosaur train. Cannot recommend it enough. Dinosaurs, crocodiles, and birds are more related to dinosaurs than crocodiles are to modern reptiles. Fun fact. Okay. Let's talk about species for a second. I just want to talk about the different definitions of the species. Okay, so the biological species concept defines species as groups of interbreeding populations that are reproductively isolated. So restricted to the definition of those organisms that undergo sexual reproduction. Do all organisms produce sexually? No. What about bacteria? The using interbreeding as a species it's not great. It doesn't really work out. I mean, again, you've got little tiny dogs like a wiener dog and a Great Pyrenees, right? Is that going to work out? I don't know. I'm not sure that. It would probably depend on who the mother dog was. I'm not sure that's even going to be possible. Are they a different species? I don't know. This is not a great definition. I don't always love using the ability to interbreed successfully as a definition, but it is the biological species concept of breeding. The phylogenetic species concept is a species in a population or set of populations characterized by one or more shared species. So the biological defines species by their ability to interbreed, while the phylogenetic defines species based on a shared evolutionary history and a distinct genetic lineage, which is just a little bit more useful in 2025 and can encompass things that reproduce asexually. But more or less, it depends on what you are researching, what you are doing, how isolated your species are. But just know there are two different definitions for a species, right? One biologic, one phylogenetic. Mendelian inheritance and its modifications assume that genes are transferred from parent to offspring, right? That's what Mendel and his little peas were doing. However, we can say that there, especially in bacteria, is a lot of vertical gene transfer, or that is parent to offspring is vertical gene transfer. What we are talking about is horizontal gene transfer, HGT, genetic material between unrelated species, mostly bacteria. So here's some past and current horizontal gene transfer that is happening. So you've got viruses, bacteria, fungi, and plants. You've got recipients forming a almost symbiotic relationship that then makes them one species. So you will have the donor gene. It will incorporate the into the, for example, insect genome, and it becomes one. The insect absorbs that bacteria. We know that this happens with viruses, right? We know that that's how viruses work. They actually insert their genome into your genome. So we've got, for example, an aphid and body color, and over time, it actually turns the aphid a different color, that particular virus or bacteria. I cannot remember. I think it's a bacteria. You have parasite wasp over here, a mosquito. It gets a gene and it becomes resistant. Yes, this is exactly what's happening here. We've got moths, we've got other moths, we've got the silkworm and the whitefly, and it's making it resistant. This is horizontal gene transfer, okay? So again, you have a donor gene from either a bacteria or a plant. The donor gene actually incorporates into the genome, and then you have the recipient genome that will turn it a different color, but mostly what it's doing is it's making it resistant. Okay, here's another example. Transfer of genetic material from one species to another. It's more prevalent in prokaryotes. Obviously, it's going to be more prevalent in prokaryotes. And the mutations and horizontal gene transfer are important sources of genetic variation. There are three primary modes in horizontal gene transfer: transformation, naked DNA that uptake bacteria. This is what we do with genetic engineering very often. Transduction, genes transferred by viruses. We know viruses do this. This is how we discovered viruses, right? It's like, what? And conjugation, genes transferred between bacteria via the pilus. This is, right, they kind of come together and it transfers from one bacteria to another. Okay, so there's three types of horizontal gene transfer. How does this happen in eukaryotes? It's much rarer. It's not very often that it happens. Mostly transposons. This is brought to you by Barbara McClintock, and the first woman to actually discover something amazing, and no one believed her. So this is transposons that transfer between rice and millet plants. Another one can be fungi uptake in the taxol gene of the yew tree. So another fungi that is making this yew tree, I think, resistant. A carotenoid enzyme transferred from a fungi to an aphid. So this is much rarer, but honestly much more interesting. It does happen. Prokaryotes do go through horizontal gene transfer, and we mostly call it transposons. Okay, now let's talk about how this would happen on a evolutionary sort of basis, and this would be what we are referring to as genome fusion. So this is the endosymbiotic. Okay, so inside a symbiotic theory of eukaryotic origin. So this is how did simpler cells develop a mitochondria or a chloroplast. So mitochondria and chloroplasts originated by the engulfing. So we know that bacteria does this, right? When you take microbiology, you know that it kind of just, if it's studying on there, it'll just sort of eat it, just like engulf it. Okay, so the enfolding or the engulfing, the eating, like a Pac-Man situation, is basically going to enfold this plasma membrane. Okay, so you have a proto-eukaryote out here, and basically it's going to just engulf it, and that's going to arise in endomembrane components. So it sort of engulfed something, and then that became the mitochondria, or that became the chloroplasts. Okay, do you see how this happens here? So here, all of a sudden, we look at a proto-eukaryote. Now we have the first, oh, I gotta go back. Sorry. The first endosymbiotic event. The ancestral eukaryote consumed aerobic bacteria that evolved into the mitochondria. This is the mitochondria genome theory of eukaryote origin. And it basically just sort of ate a bacteria that had a mitochondria that evolved ultimately into your cell having a mitochondria. And aerobic bacteria. In the second event, the early eukaryote consumed a photosynthetic bacteria that evolved into potentially chloroplasts. Okay, so you would have a eukaryotic organism that had a mitochondria and chloroplast, or you that does not have chloroplast, but you do have mitochondria. So animal and plant cells developed these organelles based on the endosymbiote, right? We know that bacteria will like engulf something. Okay, we know that cells do this. So it makes sense that it engulfed something and then sort of just became like horizontal gene transfer, became part of that process as it moved forward. Okay, again, the endosymbiotic theory of eukaryotic origin also can result in gram-negative bacteria that also has some little bit of gram-positive in there somewhere. So gene fusion was proposed to explain at least some of the aspects of gene transfer between endosymbiotes, including the origin of the nucleus. We don't exactly know, like the origin of the nucleus is still being researched, so we don't 100% know what's going on there. But again, that would just be it, ate something that had a nucleus and then just became its own membrane-bound sort of nucleus there. And also proposed is the gram-negative bacteria with two bacteria coming together, and you will have archaea that absorbed some gram-negative bacteria, and now you have gram-negative bacteria, right? So archaea, gram-positive bacteria, sorry, and now you have gram-negative bacteria. Okay, again, this is the same. You've got a nucleode here. You did some infolding. You ate it, and now you have a membrane-bound nucleus. Eukaryotes, how did they evolve? Again, this is just another picture of that, potentially to give you an additional visual of how this endosymbiosis is happening. This makes these kind of phylogenetic trees a little bit more complicated, right? It really, in reality, looks more like this and doesn't look like the pretty neat cladograms that we would like you to believe. It more looks like a web of life because you've got horizontal gene transfer mechanisms involved such that eukaryotes evolved from a pool of potential prokaryotic species. So it makes it a lot uglier than we would like to believe. You also have rings of life. Another alternative to the ring of life model proposed that all three domains evolved from a pool of prokaryotes swapping genes versus horizontal gene transfer. Just another theory. This map helps explain how certain eukaryote genes can resemble those bacteria, right? A pool of primitive prokaryotes, and it just developed like this. Only more research will distinguish this. The more we learn about genes, the more we know about these sort of things. That is it. I will get you a review video for what we might not cover in class on Wednesday.